So thanks for the invitation, and I'm happy to be here. So I said no to many years to Kavita to come, so I finally made it to come here. So after several times she invited me, I could never come. So I hope it will be worth it. And I hope to make you interested a bit in some topics which sound or are maybe not so common. I hope to convince you that they are actually common to many species and might be of interest to consider. And so what I will talk about is seed banking. So we'll talk about the fact that we'll talk about what this is biologically, but the fact that, for example, seeds can stay dormant in the soil for a long period of time, and we'll talk about which species can be affected by this. And I will today start talking about how we model this, what type of neutral model we can use. I probably prepared too much for today. It depends on the level of mathematic details you want to discuss. We can switch some things for tomorrow, and tomorrow I will talk about how this influences selection. And in my group, I must say that, so I'm a biologist by training. I mean, in France, you do a lot of mathematics nonetheless, because we are forced to do it. But most of the work I will show you is done with mathematics colleagues, right? So I don't do the derivations myself, so don't ask all the details. I took all the notes with me, but I will not do everything on the blackboard. But I would like to show you how we also build methods for inference, because that's something that now with genome data becomes very interesting, how you can use genome data to do some inference of what has happened in the past, and especially to infer this seed banking, for example. And then the last two lectures, I will move to coevolution between hosts and parasites, and I think hope to show a couple of things that we are also doing. Okay, so as I mentioned, I just mentioned once all the people who work on especially the dormancy topic and seed bank topic over the years. I work a lot with Johannes Müller, who's a mathematician, so the TUM. He's a specialist in forward equation and Kolmogorov equation, and so we do a lot of the work together. I had the chance to have a great PhD student, Thibault, who was also a mathematician from France, who developed all the SMC methods and also developed a lot of the modeling framework for the seed bank. And Diala was a postdoc who worked also on the seed bank and the selfing. And these are all the people over the years who contributed to many aspects also of this. Okay. So maybe I just recap because you've seen that last week, but just so that we all agree on what I want to talk about. So I'm interested in understanding if you have molecular diversity, if you sequence different individuals, right, in the genome, why do they look different? And of course, I'm interested mainly into neutral processes to start with, which shape this diversity, right? And so I will talk about genetic drift, but you have seen some other processes already last week. And I will talk also a bit about mutation and recombination today. And I will go into more the coalescent theory because that's what we use to do some inference from genetic data. This is I put this paper on the Slack. I find that's a paper which summarizes a lot of the current overview of how you explain genetic diversity. And I like it because it links really this ecological view with also the population genetics view, right? It's an evolution and ecology, so to say. And so the idea is that if you look at the genome-wide genetic diversity from a given species you're interested in, population or species, you see that, of course, we have mutation, right, generating diversity. What is important is the effective population size you talked about last week already. And the effective population size can be changed due to demography. So demography is changing the effective population size. For example, you can have bottleneck, so change of population size in time. It can be migration between populations. Again, you've seen that last week, right? So I'm not going to details. The mating system, especially in plants or in many, for example, fungi species, can be very important because you can have, of course, outcrossing, as you know, for many animals, right? You have two sexes, but you can have also selfing or asexual reproduction, which can matter a lot, defining effective population sizes we'll talk about later. Recombination in this respect is, of course, influenced by the mating system, and recombination, along with selection, will affect the effect of link selection in the genome and the diversity. We'll talk also about it a bit tomorrow. But what I like in this graph is also this part here, which is the life history trait. And I think that's often a little bit neglected in population genetics, but life history of the species can have a huge influence on the population size or the mutation. Right? So how does that work? For example, the life history trait, if you take the very classic ecology definitions, you have so-called R strategy or K strategy. These are different type of, roughly speaking, species, how they colonize the habitat. And so you can have the R strategy, R species which reproduce very fast, have a short lifespan, produce a lot of offspring, and they have often small propagule size. That means they have small offspring which can just migrate very fast around. And those will tend to have probably higher effective population size because they produce a lot of individuals. But then you have also some other effects which is not represented here on selection, because you have a lot of competition, you have much more offspring sharing few resources, so that generates a different type of competition. On the other hand, you have this so-called K strategy, who tend to have, for example, long lifespan, low fecundity, and large propagule size. And I think you can think of humans, right, where we have large propagule size. If you look at a baby compared to a spore of a bacteria, right, obviously. So this life history trait, here are the kind of classically used in ecology, I would say. The one I want to talk about, which is not here, is dormancy. That's why I will spend some time today and tomorrow to talk about that. I just mentioned also here that the life history trait can affect the rate of mutation. If you scale everything as mathematicians do, of course, you always scale by cell division or by generation, then it doesn't really matter. But if you observe in the population, the number of cell division and linked to the different strategy of multiplication can also matter for the mutation rate. Okay, and so maybe I start with a very simple concept of now what is seed banking? And I will use the term seed banking because I think that's how in the paper we describe it and many people use it. You will see that it's a bit more general than seeds, but let's just discuss about a very simple problem. So if you're a plant and you do seeds, right? You produce some seeds and we look what's happening during the next year. Basically, the seeds have to germinate at some point, right? And have to have the right condition to grow and to produce, ultimately to grow enough so that they can produce their own seeds later. So if you germinate too early, so this is typical of Europe weather, right? So you have the winter where often it's cold. You don't have much snow with climate change any longer, but it's still cold enough. So the plant, if you have your seeds germinating during the wrong season, they just die out, right? Because there is not resources, it's too cold, you cannot grow. But if you grow during the right season, so in spring or summer, then this is where you get the favorable conditions and then you can produce offspring. This is not what is, strictly speaking, dormancy, a long-term dormancy. This is what is called short-term dormancy in plants. It means you react to the right environmental cues. Okay, so there you don't really stay dormant for a long time. You just wait until the conditions are correct that you sense, and then you can just wake up and grow. Right? This is also true for bacteria, for example. Now, the concept of the seed bank as a strategy, an evolutionary strategy, you have to now move to a longer time scale over several years. And the idea here is that if you have a plant doing seed bank, so if the plant doesn't do any seed bank, so if all the seeds can germinate as soon as the conditions are correct, of course, you will have some seeds which will germinate in the right year. This is here the sunny year where they will develop and do offspring. Some might germinate wrongly at the year where it's cold. They just die out because the year is not appropriate. Then this one is okay. But then, for example, here you have a year where it starts really well, all the seeds germinate, and then they just die because, well, bad luck, it got cold at that point of the year, and then all the seeds die, right? And all the plants die. And then in that case, you have no more reserve because all the seeds have germinated, and that's it. Right? And so very intuitively, this is what we call the persistent seed bank, is the fact that now the seeds will germinate regularly. And of course, sometimes they will sense the cues and try to germinate, but some will just still resist. They will restrain themselves to germinate, and they will not necessarily all germinate when the year starts really well, right? So you sense the cues, but some of them are still dormant, so they will not react to these cues. And then you have seeds which will still be in the soil over many years, and then when the conditions come good again, at some point you still have some seeds which are able to react here and grow and build a new population. So this is what it's called a bet hedging strategy, right? So as the name says, don't put all your eggs in the same basket. So you wait and try to maximize your fitness over time, hoping that at some point some of the conditions will be correct, right? So that's the idea behind the bet hedging strategy. So this was proposed already early on in the 60s, and there are a few papers from Cohen is the first one, and several other ones where people do modeling to study this evolution of bet hedging strategy. I think the result is very simple, is that your environment has to be stochastically variable, so that if you don't know how the environment will be, it is advantageous to do this bet hedging, right? Basically, if you know how the environment is for sure, then why would you do it? Because you can maximize your germination to match with the conditions which will be correct all the time. Okay, so now I talked a bit about plants, but can you think about examples? So do you know some species, and I think there are many species out there, right? Not only humans. So what would be species which would be typically affected by such environmental conditions which are unpredictable and might have an advantage to do such bet hedging strategy? So you can think about some plant species, and you can think about other species. Sorry? Yeah, so for many plants, of course, you have different conditions, right? So for example, you can be in a desert where there is water once every 10 years. And so when you have water, you want to develop, grow fast, and then put your seeds and wait until the water comes back, or the other way around, as you suggest. You have water, you grow, but then from time to time, the ponds or the water dries out. And in that case, you want to have seeds which are there and can remain for a while, right? So these are plants. What kind of other species would be affected in this environment? You have also, for example, algae, right? So algae also typically do it. If you have lakes which can dry up, a lot of algae have this dormancy strategy where they put spores, the spores fall on the bottom of the lake, and they wait until water comes back again. Any other ideas? Sorry? In the context of antibiotic resistance. Yeah, so usually that's the one that everybody is scared, right? So basically, bacteria do that a lot. And there is a lot of bacteria, especially infecting humans, who do such dormancy. And actually, the dormancy can be activated when you treat with antibiotics. So if you treat with antibiotics, part of the population of bacteria will fall dormant. They will close themselves, do this kind of protection, or basically their membrane is getting completely closed. And that means that they are completely dormant for a while. We call them quiescent. I think if you look in the literature, people use the term quiescence. I'll maybe define it a little bit later. And this bacteria can just wait there for a while and then get basically what we call resuscitate or get active again later on, right? Sometimes they get active, the antibiotics is still there and they die nonetheless, right? It depends on the conditions. And so that's one of the, in medicine, I think now there are more and more people interested by this phenomenon, obviously, because we want to predict how good is the application of antibiotics, how should you apply it, should you maybe do it differently, should you do different cycles or whatever. Mm-hmm. Okay, any other idea? There are pyrophilous plants. Mm-hmm. They wait for fire to be terminated. Yes, so this is a fascinating example of plants which wait for some conditions, especially, for example, when you have fires, right? And so what is the reason is that basically the fire will kill off all the other plants, right? So eliminate competition, and then the seeds of those plants, so you need some special system of the fire is actually activating the release of the spores later, and then the spores will just fall on the ground where there is no other competition and can grow again, right? So that's one of the, and typically, I hope it's clear that the fire is stochastic, right? Not when humans do it, but when it happens naturally, of course, you sometimes have no fire for years, and then you can have 2 fires in a row, right? This can happen. Mm-hmm. Okay, any other idea? So we talk about bacteria, plants, algae. So there is one last group that we have not talked about too much. Viruses, yeah. Okay, there are 2 or 3 groups we didn't talk too much, but viruses. So what about viruses? Okay, but okay, so you see that now we are approaching some gray area of how you define dormancy, right? So is a virus alive outside of the host? That's already what you should decide a little bit here. I would tend to say maybe, I'm not sure if I would define dormancy as the virus hanging around in your environment, but think about viruses which infect humans. What do they do sometimes?
Like bacteriophage, they are when in the lysogenic phase, so they are dormant inside, integrated. Start rapidly dividing.
Exactly. So you have some viruses which will kill the host immediately, but some of them, they can either integrate in the genome and wait until whatever conditions is activating them, or they can even go into the body and not necessarily integrate into the genome, but just can stay dormant in the cell and wait. Right? So for example, you have herpes viruses in humans, they infect you, and for whatever reason, sometimes you have no symptoms for years. And then suddenly, something is triggering the activity of the virus, and then they start multiplying, then you get the symptoms again. For a while, it's very annoying, so then you go to the doctor and say, well, I can't do too much, give you a couple of things, but it doesn't really help, and then it vanishes again. But then it comes back again 5 years later, right? So these are typical viruses which have so-called the latency phase, which can be extended to very long time depending on their ability to stay dormant or quiescent. Right? Okay, so these are viruses. In the same type, some bacteria also do it. So do you know any bacteria which is very known in humans, a known pathogen of humans doing dormancy? Sorry? Tuberculosis is one. There is anthrax. If you remember, there's people who were sending at some point some anthrax infected spores to people, right? The anthrax is one of the pathogens which can just dry up and wait super long time until infecting someone. And tuberculosis is the one that, of course, many people worked on the most. There are also fungi. A lot of the fungi, so infecting humans, but also infecting animals or plants, also do such dormancy, right? So they can have the dormant stage, especially in Europe, for example, in crop pathogens. A lot of the crop pathogens and fungi have exactly this dormancy because at the end of every year, right? So you have the crops during the season, especially cereal crops, and at the end of the season in the summer, you harvest the crops, right? So there is no more crop for the pathogen. So the pathogen do these dormancy stages, which are spores, which have thicker membranes. So they are special type of spores which are produced just before the winter for overwintering, right? And usually they fall on the ground and they can wait there for a long period of time until the next season crop is available again for infection. This is also true for crops in tropics, where you have many of these fungi who also do different types of spores during the year, which can resist longer time and can be dormant for a while. And so either in the soil or for example, on crop residues or something. Okay, so that's for the fungi. And what is the last group of animal we didn't talk about, which can also do dormancy? So protist is one, and another one a little bit bigger. A little bit bigger. We can go through the whole kingdom of, so nematodes, right? Helminths. So all little worms. There are a lot of worms which can do dormancy. Actually, funnily enough, C. elegans, which is one of the molecular biology model system, does not do dormancy. So of course, that's why people probably use it, because it's useful. You can have it in lab multiplying all the time. But actually, many of the worms do dormancy. So for example, you have many diseases, especially in Africa. There are a lot of diseases where worms hide in the soil, are found in water, and can infect humans. And they have these cysts, which are basically resisting dormant stages, right? And they can resist there for also a long period of time. So what I want to show is actually that if we move beyond human biology, right, there is a lot of species out there which do dormancy. Right? Now, in humans, there is one case, and I think if you had looked at the paper I put on the Slack, there is one interesting case of dormancy in humans. So I don't know if someone has looked at it. If you have not, you can read it and you will see. And that explains, I think, now there was a whole renewal in interest in dormancy in human recently. Cancer. So it has been shown now that some type of cancer cells actually do some kind of dormant stages. And so they kind of hide in the body somewhere and wait to start a metastasis later on. Right? And so you can have this lineage of cancer cells which send some type of cells, they will stay dormant for a while and then can get up and then do a metastasis somewhere else where the initial metastasis is developing and can be treated. And of course, then when they are dormant, you can treat them as much as you want. It doesn't do anything, right? So the treatment are very ineffective against these dormant stages. So I think that has been, I would say, in the last couple of years now, one of the main interests of people to look at these dormancy models, because it has been shown that many cancer people don't understand. You can do a lot of modeling and you don't understand why you have this kind of recurrent cancer which come back again. Of course, you can argue it's because maybe some of the mutations come again, but when the people have the genomic data, they cannot find that. And I think one of the explanations is probably that you have cells which were hiding somewhere in the body, were resistant to the drug treatment, and then once the drug is over, the treatment is over, they can reactivate again.
So you're getting ahead
Yep.
of what you're going to say, but what should I expect for the effective population size?
I talk about this.
I know. What should I expect?
I don't know. Can we have an intuition? So the main effect, I'm not sure in which order I put. Okay, so I just have a slide, I think, summarizing this in a second. So that's the paper I put on the Slack, right? You will see that Jay Lennon is basically, I would say, one of the main researchers working on dormancy. He's working in bacteria, but I think they also have some connection to cancer research. And Jochen Blatt is a colleague from Frankfurt who is doing a lot of mathematical models. And I will direct you to his papers for the math and the stochastic part of it if you want to look at the demonstration. I'm happy to discuss about this, but I would not do deep in the lectures because this is too much stochastics for my taste already. So I need a lot of time to follow these papers in general. Okay, so what is the intuition behind it? The intuition is that this bettaging strategy, so do you find them in many species? I forgot to mention one of the main system people use also to study that is called the Daphnia. These are little crustaceans that you find in lakes. In Europe, all the lakes have it, right? So if you swim in there, you have them everywhere. They just don't do anything, right? They're just basically there and the fish eat them. And this Daphnia, they do this dormancy stage where when they make the eggs can fall in the sediment of the lake, and either they can be reactivated immediately, or they can just pile up the sediment, and then the egg fall dormant. And then when some fish come and then they dig out the bottom of the lake, then the sediment come around, and basically the spores can just activate and then do a new individual again, right? Or these eggs, basically. So that's one of the most known system where you can also study it in nature and do a lot of measurements. So that's quite nice. Okay. Oh. So the idea is that the seed bank, and I think that's exactly the point, is that the seed bank, they will promote so-called storage of diversity, right? Because you store individuals away from the active population, basically you can store this diversity for a while, and that means that you expect that it will increase diversity. It will increase the time lag between what's happening in the population, which is the active, and these dormant stages, right? Because you freeze, so to say, the genetic diversity for a while. Right? And so you have some part of the population which is dormant and is not on the genetic drift, basically at that moment. And that's what I will show today, is how that works and how you model that and what are the kind of consequences in the genome. Okay, I just mentioned some plants we work on because in my lab I got interested into dormancy by studying this plant. This is a wild tomato species. You can see that I just put some real biology slide. Then afterwards it will be not much, but this plant, they live into the southern part of South America. So this is Peru, this is Chile here, and here you have the Atacama Desert. So this plant, they live around the Atacama Desert, where there is really, as you can see, not much water for most of the year and for most years. And so this plant species, how did we have the idea that they do dormancy? Is because if you sequence them, you find a lot of genetic diversity. So it's an enormous amount of genetic diversity, even though when you go to collect plants, you find only 10, 50 plants in one place, right? And so to our surprise, when we sequence them, we find that the effective population size should be on the order of 100,000 locally, even though when you go out there, you find only maybe 50, if you're lucky, 100 plants. Right? So clearly, the population size that you see, which what we call the above ground, right, or what is called the census size, that means the plant you can count, is much lower than the effective population size that you can measure from the genetic data and from the diversity data. Right? And so that's the indication in this case that they do dormancy, and indeed we know that the seeds can stay for many, many years in the soil and wait until conditions are favorable to germinate again. So just a little bit of biology here. Okay, and I just put, I didn't put this paper on Slack, I can put it for people interested in more humans disease. This is one of the first paper where people discuss this evolution of dormancy in parasites. So they do different, this is a model of adaptive dynamics where they use an epidemiological model with hosts and parasite to show what would be possible strategy for the parasite and why parasite would evolve dormancy. And the idea, I just show you one of the graph. The idea in one of the most obvious case is that as a parasite, it is advantageous to evolve dormancy if your transmission and the number of susceptible individuals varies over time. That means here the transmission is a cyclic phenomenon. So you cannot transmit really well all the time. If you think about some parasites which do that, is, for example, malaria, because especially in Africa, you have this raining season and dry season. And during the rain season, you have a lot of mosquitoes. So the malaria is transmitted really well. And actually during the dry season, much less, because you have almost no mosquito left. And in that case, it has been shown that in humans and also in the mosquito, the malaria has some kind of dormant stage where they wait until the rainy season starts to activate and get transmitted again. And so it also explains in part this relapse when you have malaria in your body, right? That sometimes you just have nothing for several months, and then suddenly the disease starts again and you have this relapse with the symptoms. And that's probably due to this adaptation to this kind of cyclic transmission. I must here be a little bit cautious because technically speaking, I think the malaria, so the classic one, the Plasmodium falciparum, is not really doing dormancy. It's more some kind of, let's say, call it a resting stage, but it's within a year, within a few months, right? And this relapse never lasts too long. There is another malaria parasite, which is called Plasmodium vivax, which is found in South America. And that one is known to do real dormancy. So it can stay really dormant for several years into the body. It hides into the human body, into the liver, and then can reactivate much later on. And we don't really know why it can get reactivated or not. So the falciparum, I think, is not the real case of dormancy, I would argue, and it's not a real bettaging strategy. It's more an adaptation to the seasonality. I think the vivax seems to have a real bettaging strategy here. Okay, so I didn't put this paper. If you're interested, I can put it. It is an extension of this latency phase I was mentioning, that some viruses or some parasites have this long latency period, right, in the body. It is an extension of that, so to say, but the mechanism are a little bit different here. Okay. And maybe I can explain the difference between quiescence for when you search in the literature. So how we define it, and I think that's now, I would say, how also Johan Blatt and Jay Lennon define it. Quiescence is the fact that, especially for parasites, but also for whatever other, especially for bacteria, is the fact that, for example, you have one individual. This individual can be dormant itself, right? Stay dormant, then get activate again, be active, maybe potentially fall dormant again, and then reactivate, right? But it's the same individual, right? So there is there's the same bacteria or the same spore which can switch states. In the dormancy and what we call seed bank, we usually consider that then the offspring are dormant, right? So like in a plant, so you plant above ground, you make seeds, and your offspring are dormant, and then at some point will wake up and make a new plant. So it's a little bit of a semantic, because in the models it doesn't matter, but just sometimes to define things a bit more precisely, one should be careful. And in literature, you will see all these terms all the time, right? And especially in humans, it's even worse, because people invented this term of covert infection in human parasitology, but that's exactly the same. Right? So I'm just mentioning all this, but I will now call it seed bank or dormancy in general. Okay, and just to close, for parasites, what you can show is that, for example, if the transmission, the disease transmission is stochastic or variable in time, you will favor quiescence. It can also work if you have superinfection, which means, for example, if you're infected already by one parasite, and then you can get infected by several other strains in the same time. If the first strain, by being dormant, can protect you against being further infected, then also dormancy can evolve, right? Because basically for the parasite, it's advantageous for some strain to be dormant so that they actually prevent that other strain will compete into the body with this resident strain. What we have shown with the student of mine is also that if you have coevolution, so on a longer time scale when host and parasite population will change in time in the genetic composition, I'll do that on Thursday, but you can also show that this favors also dormancy. So it can be advantageous for the host or the parasite to stay dormant, so to say, because then you wait until there is another cycle of coevolution where your allele will have an advantage, and that works because you have this frequency-dependent selection that we just mentioned early on a little bit as well. Okay, and so what I want to show now is that what is the effect of dormancy, and we'll look a little bit through it through different models. So this is a slide summarizing more specifically for plants, but that's true also for fungi, algae, nematodes, and so on. And I just copied the idea of this graph I showed you before, but now integrated here what is specifically relevant for the effect of dormancy. So if we want to explain the nucleotide diversity that we observe, right, in red are the boxes of processes which are affected by the dormancy. And so the one I will talk about, of course, is the effective population size here and the effect of genetic drift, right? How does that work? It works that you have a census size, so the number of individuals which we will call active or above ground for the plant, right? And this numbers, of course, is mitigated by the fact that you have seeds, which will germinate and replenish this active population. So, of course, the dormancy affects then the seed availability, so how many seeds there are and how many will germinate. It also influences extinction recolonization, because if for whatever reason your population gets extinct, if you have these dormant seeds, they can recolonize immediately this patch, right? And so in plant ecology, this is extremely important, where you see that many of the species which are present over a long period of time in different habitats, they always have this replenishing by the seeds. I think the mode of reproduction and so on I just mentioned before in the other graph are not particularly affected necessarily by the dormancy in this case. The mutations can be affected. Why? Because it will depend if you're dormant, are you having mutations or not? So there is a big debate. I'll talk about it later. We can also discuss about this later, whether in the seeds or in dormant stages, mutations happen. Right? And I think people disagree on this. I think as far as I would say, there is not really good biology, genetic data at the moment to assess this particularly. I think nobody really has tried to understand it. But it is important, right? Because if you have this long dormancy stages, if they also mutate, especially if you think about bacteria resisting antibiotics, if the dormant stage of the bacteria is also mutating, it can also create a lot of genetic diversity. You can have new mutants appearing, which might be even worse than before you apply the antibiotics, right? So that's, I think, very important, even though we know very little about it. Question? Yeah. Yes, so the mode of reproduction is basically whether you do, for example, selfing, so if you can reproduce with yourself, or if you do outcrossing where you need a partner, right? And this affects effective population size because if you, well, I talk about it in 2 days. So, but the idea is basically if you do selfing, you have less, the effective population size is decreased because you have more genetic drift, because you reproduce with yourself, so you produce more homozygosity over time, and it decreases the genetic diversity of the population, compared to if you have always 2 individuals who have to mate to do sexual reproduction. That makes some kind of sense? I show a bit more on Wednesday. I didn't want to go into detail today about that. But you didn't talk about selfing last week? No? Okay. Sorry, plants are weird. They do all sorts of weird stuff. Yeah.
Is the basis for dormancy is purely genetic in nature, or is it also something that phenotypic layers or plasticity?
So I think that's a good question. That so obviously they are genetic determinants, right? I think the question is, what is the trigger for dormancy? And I think there it depends on the system. It depends, I think, on the species, I would argue. I think the idea, I didn't put this model actually, that would have been maybe easier to explain that. The idea is that if you do no dormancy, right, and the environment is stable, you might, okay, that's true that Sarah showed that last week. If you look at this equation with the selection, the fitness of the population is maximized, so selection will push to maximize the fitness of the population. Right? And at that moment, all the individuals have the maximum fitness, meaning that actually you don't do dormancy if the environment is stable, because you maximize your immediate fitness. And that only works, and dormancy can only be advantageous if you cannot maximize your immediate fitness because the environment is too variable. Right? And so how does that happen? I think it depends. So in bacteria, they seem to be very sensitive to chemicals in the environment, for example. I think the plants much less. I think the viruses are obviously depending on when they enter the host and what's happening inside the cell where they are. I think malaria seems to be also detecting which cell of the body they are entering, and obviously seems to direct themselves to enter some cell of the body where they can do dormancy. So it is a plastic response, obviously, but I think how it is coded, I think we don't know, at least at the biology level, I think not much is known about the pathway which are involved there. In bacteria, a little bit, but for example, in fungi or malaria, I don't think it's really well known. And in plants, obviously, they do a structure, right? The seed is a complete structure where you have inside the embryo, it's protected. So it has evolved since millions and millions of years as a strategy to protect the embryo, right? And in plant, I have to say that you have huge differences between species. If you take Arabidopsis, for example, it doesn't do any dormancy. So the seeds fall into the soil, they germinate immediately next year, and that's it. But you have other species which invest a huge amount into some dormant seeds, right? And seeds can resist for a long time. So I think it was a palm tree. They found a coconut. Yeah, they found a coconut which was, I think, 4,000 years old and still able to germinate. And so obviously some plants put a lot of effort into make sure that one seed can stay for a very long time, still potentially germinating. Yeah, okay. And so there I will not talk much about this, but I will talk about selection a bit tomorrow and recombination. And so we can discuss about how it affects link selection. But that means that as dormancy will affect the strength of selection, it will potentially affect also the effect of selection in the genome and diversity. And so it gets a bit more complicated, but I'll try to touch upon this tomorrow a little bit. Okay. And so what is the main idea of how dormancy works? So I just mentioned this, just for, if you do plant biology, I think now in conservation biology, this is really assumed that dormancy has an importance, right? So if you do conservation biology of plants or some animals, this is very important. I'll talk about that tomorrow, about selection. Another aspect I will not talk about is how it links to different type of selection, especially balancing selection or coevolution with parasite. that also there are many effects of how dormancy affects here how selection happens. It can maintain cooperation, which is also an interesting aspect if you think about, for example, bacteria systems where you have also this kind of cooperation and competition. The dormancy can affect and can announce the maintenance of cooperation, for example. Okay, and what I would like to do today and until Wednesday is to model a little bit quiescence and dormancy, and we would like to detect their existence. And as I'm a fake biologist and don't like to do experiments, I would like to just be lazy and use sequence data to do that instead of doing very complicated experiments. Right? You can see that if you want to prove dormancy, it's very hard, right? Because in the lab, you have to do experiments where you have to make different germination. So for example, if you have seeds which need a lot of time to germinate, you have a problem, right? I think there are some colleagues, I think in Germany, who started an experiment of bacteria dormancy. They want to make the bacteria dormant up to several hundred years. So they are planning an experiment that they hope someone beyond their lifetime, and I hope their institute will not get dissolved, right? They plan that at some point someone might be able to see this bacteria which have been sleeping for 100 years, right? So you can see that this is a little bit of a problem for doing research on this because, well, we don't do research at this time scales of a PhD student is not 100 years, so obviously this is a bit problematic. And so what we try to do is to use sequence data to study that. So I put a couple of review on the Slack. So this is the mathematics has the main, this one has the main results of the mathematical models that are used. This one has the generality and a bit of the basic principles, and that's the one I showed you before. Okay, so now I was not sure what you have done exactly last week, so I will skip a couple of things or go through some of this concept reasonably fast, I think, to move to the seed bank at the end, right? But if there are some things you're not sure, just scream or say something, or we can stop and do it later also, right? And I think you will also have some, again, so I don't want to give all the results now. Okay, so I'll just do a little recap of what we need to know to understand how the seed bank model works. So I'll talk about the Wright-Fisher model. Wright-Fisher model is a very simple model of population genetics. I think you've done that, right, last week? So I will not go very long into this. You have some parents, they produce offsprings. Then the offspring choose their parent at random, okay? And either you define it with n diploid individuals or 2n haploid or n haploid, depending on how you want to propose it, okay? And that's how it looks like, right? So you remember that you have some parents, you have some offspring, you have 2n parents, 2n offspring, and so each of the offspring choose one parent at random. And in fact, here we track chromosomes or loci, depending how you want to see it. Okay, and so that's the untangled version. And as it is a neutral model, all the individuals have the same fitness. That means I can have exchangeability of all the individuals, whether they're in position 1 or 2 or 3, doesn't matter. So I can reorder them, and here I get these lines which are not crossing any longer, right? But that's the same. So you should scream if I say too much, right? Which you want to say. Okay, and this property is called exchangeability. So you can exchange all the individuals. There is equal probability to be picked as a parent. And I think you saw last week already that what is the probability to be picked is 1 out of 2n if you have 2n individuals, right? Okay, and so that's also the probability which is called the probability of coalescence. That means if I take 2 individuals at random, at one population, what is the probability that they choose the same parent? It's 1 out of 2n, right? Because one individual chooses a parent, okay? And then the second one has to choose the same parent. And you have 2n parents, so you have 1 chance out of 2n that this happens. Does that make sense for everybody? Okay. This is here the formula of the binomial sampling. If I want to know the probability of producing k offspring, right? So that's the probability that a parent produces k offspring in this model. And so you have to choose 2n in k, 1 out of 2n is here, and 1 minus out of 2n. Okay, and this you can also approximate with the Poisson. Sometimes you see that I will not use it, but it is often used in many also textbooks or papers that you can approximate with a Poisson distribution. Okay, so that's how it looks like. Concretely speaking, if you go from the right Fisher to the discrete coalescent, what is the relationship is the fact that if you do the right Fisher, it's forward in time, of course, but it's a bit inconvenient if you have sequence data, because the sequence data we have, they come from the present usually, right? So we have individuals that we can sample now, and we want to know what happens in the past. So that's what we built as a here sample of little n. Usually the sample is little n, and all those little n individuals, you track the genealogy of those individuals. So to say, that's the principle behind the coalescent theory. Okay, so we have n samples, and this n samples at some point find a common ancestor. So you can see here there's two lineages, they find a common ancestor here. And then again, these two lineages find their common ancestor here, and that's the most recent common ancestor. This individual is the most recent common ancestor of the sample. Okay, and if you sample enough individuals from an empirical point of view, you want that your most recent common ancestor of your sample is the same as the most recent common ancestor of the population. So that means that you have more or less a good idea of the genealogy of the population. Okay? I think June will explain a couple of things about how you get some statistics about that tomorrow, right? Or yeah. And okay, well, that's what I just said. Yeah, so I just removed those individuals here. Just the idea of the coalescent is that all those individuals, you have no idea about them, right? Because they are not in your sample. So if they are not in the sample, you have no information about those individuals, right? Okay, so that's how you get to what is called a discrete coalescent model or discrete time coalescent. And you just use this probability that two individuals share a common ancestor. As I said, it's 1 out of 2n. The probability that they don't share one parent one generation ago is 1 minus 1 out of 2n. Okay, and then you can just write very simply for two lineages, so for sample size of size 2, then we can calculate the probability that two lineages coalesce t generations ago, so they have coalesced, and then for t minus 1 generation, they didn't. Okay, so that's just a recursive equation where you just write, or they didn't first time, twice, and so on, until t minus 1 times, and the last time they coalesce. Okay, I think you've done that also last week. So, and okay, that's what you get. And this distribution is, or this probability is basically exponentially distributed. That's a geometric function. And so you can use the fact that the geometric function can be approximated by the exponential and write exactly here the probability that two t lineages coalesce at least t generations ago. So this probability x is bigger than t, bigger or equal than t, is 1 minus p to the power t, with p is 1 divided by 2n. Okay, so that's the idea behind the discrete coalescent. I think I do that in the biology class, and so I thought that was probably more appropriate for most people, I think, to get the intuition, because if you do the real mathematics stochastic demonstration, I find you get lost a lot of times. So we can do it another day if you're interested, but I think that at least gives an intuition about what's going on. Okay, and as you talked about last week, this probability of coalescence is higher in small population than in bigger population, and the coalescence is faster in small population. Okay, you can extend it to a number little n of samples. Okay, so that means that now at every point in time we have n samples, and out of the n samples, 2 out of n can coalesce. Okay, and so they always have a chance 1 out of 2n that this happens. And so you have this choose n in 2, which is n minus 1 over 2 possible pairs of coalescent at any point in time when you have n coalescent n lineages. Okay, and so then the probability of coalescent is just now multiplied by this number because you have more chance that this happens. Okay, and then you can calculate the time until the first coalescent event, and so it depends on the number of sample little n, right? So any 2 of this n can coalesce, and they have a chance out of 1 out of 2n that this happens, and that's an exponential, again, distributed distribution. Okay. And from that, I skip the mathematical stochastic details, but then from this idea, you can build what is called usually the continuous Kingman N coalescent, where you scale in this process in the limit. So you rescale time by 2n. Okay, so the time now is scaled in 2n units all the time, because we know in 2n units there is a property 1 to have a coalescent of 2 lineages. And then you scale in 2n, and then you can move usually to say you have a big population size enough so that the number of samples is smaller than the population size, and then you can compute this distribution of the coalescent times. And so how does it look like? It looks like this. You can use this distribution of the coalescent times to calculate now in the tree. Here I have 5 lineages, right? Do it here. So we have 5 lineages to start with, and then I can calculate what is the expected time to the first coalescent. So how do I do that? I have 5 lineages, so that means any 2 out of the 5 can coalesce. Okay, I don't know which one, right? Randomly, I will choose some. And the time is given by this, so you have an exponential distribution, and that means the waiting time is here defined, and will be actually 2n divided by 10. Okay, because we have 5 samples and 2 can coalesce, and that's this first time here. That's the time 5, this is the expectation. Now, at that point, we have 4 lineages left, and then we have to wait now until the next 2 have coalesced again, right? And so this is what is happening here. We have this time, expected time from 4 lineages to 3, and it will happen, for example, here, where we have these 2 lineages coalescing, and now this time is on expectation 2n divided by 6. Why? It comes from this choose n in 2, right? So basically, every time when you have 5, you have 5 times 4 divided by 2, so you divide by 10. Then you have, again, here, 6, because you have 3 times 2 divided by 2. And then the next one, we have now 3 lineages. I wait again until they coalesce, and here we have 2n divided by 3, that's the expected time. And then the last, we have 2 lineages, and that's what you've done last week, I think, where this time is expected is 2n. Okay, so I'm just extending the formula that you've seen last week, right? I think. So now 2n lineages. And what we can do is we can calculate here, and I'll just do it, but June will use it tomorrow. So you can calculate the expected length of the tree, which means I want to calculate the total length of all the branches, right? So the total time which is in this tree. So I calculate the sum of all branches, right? So the total time. And so what is it? You do the sum of this one. So you do this here times 5, because there are 5 branches, right? Then plus here we have 4 branches times this expected time. Then we have 3 branches times this, and 2 branches times this. Okay, so that's the formula that I wrote here. Then you can rearrange that, put 2n as a factor, obviously, because it's the classic factor of the coalescent. And now you can rearrange this formula to obtain this really nice sum here, which is 4n times the sum of 1 over I. I is 1 to n minus 1. Little n is the sample size. Okay, so that's how you obtain on expectation here the expected total length of the coalescent tree. And I will just say as a teaser that it is useful, as you will see tomorrow, to know this formula. This you didn't do last week, right? Or, okay. Now you can put mutations on the tree. Again, you talked about it last week. So you can put mutation. We usually call this mutation rate mu, right? And this can be the mutation rate per site, or it can be per locus, depending on what you consider, right? Or per genome, whatever you want to define. Again, I think this you've seen last week, so I don't want to go into detail here. And again, what is interesting is, of course, that you can at each generation define whether there is mutation or not. And that means that you have, again, an exponential waiting time for mutation to happen. And you can use that on the tree, because you can go along the tree and say, if I have now an exponential waiting time for coalescent times and exponential waiting time for mutation, I can get some ideas about the average expected number of mutations on the tree. And that's what you can do, here, where you can calculate the total amount of segregating sites, which is called big S. And this is the mutation rate times the expected length of the tree that I just did. And then you can get this formula where we have now this theta, which is the population mutation rate appearing, as you have seen last week, right? Okay, everybody remembers that? Okay, so the additional part I would like to talk about quickly is recombination, because then I don't have to do it on Wednesday. But it's also important when we look at genome data. So just a little recap. I think, again, you have done that last week, maybe not in the coalescent framework, but in general about recombination. I remind you that the recombination happens when you have crossing over, and then you move pieces of chromosome between chromosomes, which when they pair, right? So we have here some parental chromosome, then they will recombine, and you have exchange of some pieces of chromosome between them during the meiosis, and then you produce by meiosis 4 haploid gametes, which are all pieces of the different chromosomes of the parent. Okay, so I think this you have done, so I will skip it. These are just the fact that either you have free independent segregation, it's called the Mendes law, where if you have two loci A and B, here we have big A or big B allele and small a, small b alleles, and then if you recombine them in an F1 and then you cross the F1, you get 25% segregation of all the possible genotypes. If you have linkage disequilibrium or non-independent segregation, then you might have an excess of some of the association, right? And that's what you've done last week, and Sarah explained, I think, linkage disequilibrium. So, okay, so that's the recombination rate. Now, in the framework of the coalescent, the recombination looks as follows. What we can define is the probability that there is a recombination event happening, and we call it usually little r. Right? And you can define the probability that two loci have the same parent with the probability 1 minus r. Okay, and that we will define, I'll show you in a minute, this effective recombination rate, which usually is called rho, which is 4 NER, and you can see it's very similar to the 4 NE mu, which was the theta before. And yeah, okay, so I skipped a bit of the demonstration here, but the idea is that we have this recombination event happening in time, and you can have, again, the same idea that you get a longer lineage, the probability that you recombine times the probability you don't recombine for some amount of time. And again, that's exponentially distributed. You can again approximate it with the exponential distribution. And you can find now that, I put confusing slides here. Let me just go here. And this is how it looks like if you track back now your genealogy of two samples. So we have two individuals here, A and B, right? And when we look at what is happening by recombination in time, so you have now two events which can happen. Either you have coalescent event, so you find the same ancestor, right? Or you have recombination event. And recombination event backward in time, that's the confusing part, is actually splitting. So now your lineage is just splitting into two instead of merging as the coalescent event, right? So it's the other way around. And why this is? Because here this recombination event means that I took one gamete with this part of the chromosome here, which got recombined with this gamete with this part of the recombination, and they fuse forward in time to make this. Okay, so forward in time, it's intuitive. That's the meiosis I showed you before, right? You have two pieces which are put together. So backward in time, it means this piece is splitted. So you split along a lineage the two pieces of your locus, and they have their own, again, coalescent independently until at some point everybody merges to a common ancestor, which is the common ancestor to this locus, and it has the piece which you find in every individual. Okay. I don't want to give much more details up to now. We can discuss about again if there are more questions. I will talk about it on Wednesday again if needed. Are there some questions so far? Okay. So now I'll do the same, but with dormancy, right? So it's just very simple. So now we'll just do exactly the same kind of ideas using dormancy and with a seed bank. And so there are two models which I used. I think I prepared a lot of details, which I probably will not go through all of them, and depending on how much time and questions we will go through. So the first model is an earn model from Kai, Krone, and Lascaux, which is the first model of seed bank in the coalescent framework. And that has been very useful, but it has some assumptions, and that's what I want to discuss a little bit with you, how it works and what are the assumptions behind it. And then the second model is the seed bank model by Blatt et al., which is based also on a different principle, and all these two models, they give different results and different expectations. And that's why it's a little bit interesting to compare them and discuss a bit which one should be used or not. Okay, so let me just go through the model of the Kai, Krone, Lascaux model. Actually, I was planning to draw something on the board, but I can do it like this. So the idea is now we do a kind of a Wright-Fisher model, but we have to account for dormancy. So what does that mean is that if you go forward in time, I'll still draw something. Can you see here if I draw something here? Is that right? So if you remember in your Wright-Fisher model, right, we have some individuals, then we have the next generation. Okay. And the idea is that, of course, you have some individuals who will give offspring to the next generation. That's the classic Wright-Fisher model. But what is happening with the dormancy is that maybe this individual here will make some seeds which will remain dormant and give only this offspring, right? It will skip some generations, right? And then maybe this one will also do one which comes here, and then maybe this one, whatever, and this one as well. But those ones will just do the classic, right? And all the other ones can do the classic, for example. Okay, and so how do you represent that? You see that when you start doing this line, it gets a little bit complicated. So what you can do is you can do an earn model. And so in this earn model is basically you have your population, but instead of putting it on top of each other, you put them next to each other, right? So this will be generation 1, 2, 3. And so here we have the 1, 2, 3. And so you progress in time. And so you can say, okay, if I take these individuals which are in this generation 1, in the Wright-Fisher model, that means they will make immediately offspring for the next generation. And so maybe this one diffuse, they have the same parent. Okay, so the individuals of that generation make offspring for the next generation, and then you move, and those ones will go to this, and so forth, right? Now with the seed bank, what you can do is here, that you can see that you move the individuals, but now these individuals, they actually can move much further because they can just stay dormant for long, right? So basically, instead of going to the next generation, they can just say, well, no, I stay dormant for M generation, okay? So that's the idea that you shift now over several years or several generations in one go. Now, I have to say that that's the forward interpretation of this model. Actually, this model is a backward in time model. That's a coalescent model. So it's meant to be the other way around. What we assume is here, this population 0 is the present day sample. Okay, so that's the plant you can see. You can collect them out there. And then they come from some individuals in the past, right? So if you're in the Wright-Fisher model, basically those guys, they come from this population, only this, but now they can come from 1 generation ago to up to M generations ago, right? So this here is a seed, is a plant which was made from a seed which was produced M-1 generation ago. Okay, so you just go in time. Yep.
If you can have parents from like skipping generations, how do you even define a generation here?
I would like not to answer this question right now, but I think that that's exactly one of the questions. So I think in a way, the generation, what is a generation is now rescaled by this dormancy time, obviously, right? So if you want to define a generation, actually what we have to say is now that you need to account for everybody all this time that you can stay dormant as a generation, because the definition would be that all the offspring are replaced. And that's the definition of a generation, that you start from parents and you wait until all the offspring, all the parents have been replaced. And of course, in this case, you're right. it means that we have to wait a long time and not only one generation. As in the Wright Fisher model, we have to wait several years or time steps, you want to call it, until everybody has been replaced. And so the generation time in the seed bank, when you have dormancy, should be accounted for that. The problem is how do you know it?
This is like Wright Fisher, but with science. Parents sort of with some
Yes.
like random
Yep.
time between.
I discuss about the distribution in a second. Yeah, exactly. And so that's why it's a little bit, I think if you talk to biologists or mathematicians, people have different view of how you should define the generations, right? Because what is a generation here, especially if you work on many species, we don't know this dormancy time, for example. So what should I know what is a generation? Very basic example, there are bacteria which can remain dormant for 100 years, right? 100 years. They multiply generation time in the lab is half an hour, 1 hour, let's be generous, right? So what is the generation time now? Is it 100 years? Is it 1 hour? Is it 1 hour times whatever to get to 100 years? I don't know. But in the lab, usually you do it in the lab, you have your bacteria multiplying immediately, right? So you don't have this bacteria you keep for 100 years. But you go in the wild, and that's what you will find. So what is the generation time? Depends, I guess, what you're interested in measuring at the end. But the official definition, I would say, from the model would be that you have to wait until everybody is replaced. And that takes, of course, a lot longer now, a lot more time steps.
But if I have 10 individuals,
Mm-hmm.
all n will be updated
Yes.
in time for order n.
Yes.
So if m is small enough,
Yeah, I show how it looks like exactly. You can compute it from there. Yeah. Yep.
Is the probability of picking up here that goes back n of these
Mm-hmm.
slots, is it exponential?
At the moment, I don't assume anything. I'll do that in a minute. But at the moment, let's say that we just define this probability B, which you can call BI, right? And I would be how many years ago this plant comes from, right? This seed come from. So we have here B1 from one year ago, 2 years ago, until M. And you can see here what is in this model very important is that we have so-called, they define here in this paper an M window, which is the maximum amount of time that the seed can have been dormant, right? So that's a biological kind of threshold to say, okay, the seeds can stay dormant for 50 years or 100 years, but not more, right? At some point, it's not possible. And so it has a boundary here. And now you can define this distribution here of the germination rate, right? So I just mentioned it again. So here we go backward in time, but forward in time, this is a germination rate. So that means this is here, this B1 is a germination after 1 year. B2 is a germination after 2 years, right? And so you can either say, I know it, I have some estimation, or you can assume, okay, basic principle, it could be geometrically distributed, right? And so that means you could have a very simple function where you say I have a probability B to germinate, and then I just do an exponential geometric distribution with this, right? That's possible. Whether it's always realistic, I don't know, right? We'll discuss about this in a minute. Okay, so the time of dormancy can be any function of time, but usually we use, of course, the geometric, but you can assume something else. And the time of dormancy of a seed is bounded to this value M. Okay, and of course, what we assume implicitly in this model is that the M will be small enough compared to the coalescent times. Right? Otherwise, you cannot derive the model. Again, I will not go into details of the stochastic. I have the paper and I took some notes if you want to discuss later or tonight. We can discuss into more details, but the idea is that this M has to be reasonably small.
that's the meaning of weak.
Sorry?
Weak means what? M is much smaller than
Yeah, so weak, yeah, so I come to what weak and strong means in a minute with the other model, because it's a kind of a difference between the two models. So weak here, it means in the sense that the M has to be small enough. And so if you take 100 years, 200 years, even 1,000 years, it's still small compared to the coalescent times, which potentially can be thousands and thousands, right? Okay. Yep.
Why are you writing
No, yeah, so I think these are not minus, right? So these are just, I think I just wrote, yeah, so this is not a minus. Yeah, it's just to show you switch. So the idea of the model, what they do in this model is you start with all your sample at time 0 in the active population, in the above ground, and then you relocate them into the different urn, okay, in the past, so to 1 or 2 whatever years ago. And then once this is empty, you slide the window, right? So this one is empty, you move, and now you start with this population again, right? And then you have these samples here, and this has to relocate to somewhere in the M previous window. Once this population is empty, you move again once, and then do the same with the samples which are here. And what happens is then, of course, that there is a moment where you will have 2 samples falling on the same ancestral individual, and that's your coalescent event, right? Because we have one seed which come from one sample, from one parent, and then we have another seed which come from the same parent, and that means you have now a coalescent event of 2 individuals finding the same parent. Okay, except that now I think it's pretty obvious the intuition is that if everybody goes from here to here, you have much more chance to find the same parent than if you start spreading everybody in M windows, right? I think that's the intuition behind it. Okay, so that's how the coalescence happens in this model, basically. And so what you can prove, and again, I put here in one sentence a long demonstration in a paper. What you can prove is basically that this model is a time rescale ancestral process. So when you take the population size to infinity, and it's the same as the Kingman coalescent, but run on a slower time scale. So what that means, we have exactly the same process as the Kingman coalescent, where the coalescent rate in units of 2n is scaled by this k, choose k in 2, but we now multiply by this factor here, beta square. And what is the beta square? That's the formula here. And you can see that here I don't assume any B distribution for now, I don't need to, but this is the sum of the BI, so the sum of all this probability of germination. What is it? It must be 1, right? It must be, because all the sample you have now, they must have come from a germinated seed, right? So that's just a proof that they should work. If you sum all the BI from 1 to M, you find 1. And here you have the sum of the IBI, and what is this? This is the expected amount of time that the seed will spend in the in dormancy, right? So how long you sleep on average, and this is what usually also we call big B, this function here, and this we call usually beta, which is basically at the end 1 divided by B, and that makes sense. What is interesting in this model is you can see that actually doesn't really matter on what is the distribution you choose for the B. So that answers your question now back, is actually whatever you choose a germination, geometric function or not, doesn't really matter. What matters is the average time that you stay dormant in this model. Yep. Sorry, say again?
So all the individuals come from dormancy.
They come from a seed.
But need not be dormant.
So maybe I phrase it a bit differently. If you come from this guy, that means this seed has been 1 year dormant, which means basically it wasn't dormant, right? But then all those are dormant seeds which were dormant for 2 or more years.
The same way that of one,
Yeah.
the other individuals which came from other seeds.
Yeah, but we don't, so that's our sample, right? So your sample is only the one you have sequenced, you have. And this we assume for simplicity here that they all come from the present population. Actually, you can do the same. The model also works and the same result apply if you would sample also some seeds. So you go, you dig out some seeds, germinate them, sequence them. That means you have samples everywhere, right? In the past, potentially, and it works exactly the same way. Right? So the model is very robust to that. Actually, you can show in the limit, it doesn't matter. I think in the paper, they showed that case for the demonstration, but I think you can extend it to more and it doesn't matter. One of the limits, don't forget, I mean, that's why I highlighted this before, your sample size should still be smaller than the population size. And here the population size is all this, right? So it's the M generation in the past. Right? So you still can sample a lot. You're still safe that you will be smaller, but you cannot sample all the seeds, because if you sample all the seeds, then you tend to be in a model where you violate the assumption of the classic coalescent, because your sample size is too big compared to the population size. So that is not allowed here, at least in the derivation. Okay, and so that's the intuition. And so why is it a beta square? I think that's the main intuition to get from this. So why do we scale now the coalescent time by beta square? By the way, beta is between 0 and 1, right? So if it's 1, that means there is no dormancy. That means everybody germinates immediately. So we are back into the Wright-Fisher model. And then if beta gets close to 0, it cannot be 0, obviously, but it can get very small. And that means beta square, it can be very big, right? I mean, and so why is it beta square? Why is it not beta? So beta is the average time you spend in dormancy. So I think it's good because I don't have to teach too much, right? So it's very simple. Obviously, for two individuals to find the same parent, they must come from the same parent sometime in the past. That means actually that both of them must have come from the same ancestor, and actually that they have the same probability to have germinated, which is beta each. So the two lineages have a probability beta to choose the same parent, and then beta to choose again the same one. And that's the difference to the Wright-Fisher model, where you don't have this because you choose 1 out of 2n from the previous generation, and that's it. But here we choose beta, where do we land in the past, and then out of those 1 out of 2n, right? Because in each of this box, you have 2n individuals in the past, which were the plants which were living. Okay? So basically, you just extend the Wright-Fisher model now to several generations in the past, or years in the past. I keep saying generations, it's really confusing. Okay, and so that's the reason why we have this beta square. You can define the seeds. So yeah, okay. So now from this model, we also wanted to define the mutation. So what can you do? There are two different questions. Either we assume that only mutations happen when you have germinated, or the seeds can also mutate. Right? Depends what you want to assume. So you can just do a combined mutation rate where we say mutation which have are in the active part of the population. So these are the active one which are above ground, and they are multiplied by beta because of course this mutation rate is only for the plants which are above ground, which have germinated. And you have a mutation rate for all the seeds which are in the seed bank, and this is 1 minus beta because of the one which have not germinated. Right? And so that will be your combined mutation rate for everybody. And now I think that's where the assumption is very crucial. Is there mutation in the seeds or not? Because you can see that now, if there is no mutation in the seeds, I have to remove that. And if there is no mutation above ground, I remove that, which is, of course, not usually realistic. Right? But there is a huge debate, and I think we try to spend many papers arguing about different ways. I think there is no good data to, for sure, give an argument about either way. Our interpretation and my interpretation from studies we have done a bit on phylogeny and different papers is probably we should consider that there is indeed mutation in the seed bank. That's what we use classically in our model, and all the results I'll show you afterwards come from there. Right? And we assume that this rate is the same as above ground. So the rate you have in the seed is roughly similar as the one you see in the plant when they are multiplying above ground. Okay, so that's a strong assumption here that I'm not, I don't want to hide it, right? And that's why I say it explicitly, because a lot of the results come from there afterwards. Okay, and what you can do now is you can put all of these things together. And so what you can show is that, yeah, so if you assume that the mutation rate in the seeds and in the above ground are the same, so we get this mu is mu A equal mu SB, and that you can see that if you do that, you get the mu, right? And that's the mutation for any seed or plant. then it yields this new theta, which is the population mutation rate, and now it's 4 NCS mu over beta square. And what is this NCS? Is the census size I mentioned, which is the size of the above ground population, right? And this NCS, in fact, is here the size of one of those, right? So here I have NCS plant, or 2 NCS plant, in this case, okay? And so you can see now why that gives the intuition of the question of Kavita. If the theta, you have a beta square, which is very small, your theta can explode and get very big, right? Because now I have maybe 50 plants above ground. So I think, oh, you know, shouldn't be too much diversity. And then you have a 0.01 beta, your population size and your theta is exploding, right? Because of that. You can see that, of course, this beta square depends on this assumption here, and that's important, right? And so if you assume that there is no mutation in the seeds, actually you have only beta here. Because you lose one of the factor because you have a beta scaling in the mu, and so you have a 4 NCS mu beta divided by beta square, and so one of the beta vanishes, and so you have 4 NCS mu over beta. Does that make sense, or should I write it, or? Okay, so I will assume that we use that right now, and on Wednesday as well, just for practical reasons. Yep. Okay, so should I write it down a little bit more maybe, right? So if so if your mutation rate now is only in the above ground population, then you find this mutation rate, right? Because the other one is 0, because there is no mutation in the seeds. Okay? That means, sorry. Then we need to write now the theta, and it's 4 NCS, okay, times mu, which is now this. And it's still divided, it's still divided by beta square, because that comes from the coalescent. Okay, and you can see now that we have this, which means here we have 4 NCS mu A by beta. Okay, and so you can see that now it makes a huge difference because I have only a scaling by beta and not beta square, which can matter a lot, right? For the diversity, this might make a huge difference. Okay, and I mentioned an additional result, which is important, is that now if you look at the recombination, we can do the same process. It is not in this paper, so we did it ourselves in another paper. And if you follow the recombination will be now rho 4 NCS, so the same idea, little r. That's what we had in the classic right Fisher model Kingman coalescent. But now it's divided by b. So why is it divided by beta? Sorry, by beta. Why is it divided by beta and not beta square? It's not dark magic. Because recombination works only on one lineage backward in time, right? So in fact, your recombination happens when you're outside, when you have a plant above ground, which is doing meiosis, right? And actually it's only one plant. You don't need a coalescent event of 2 lineages merging, but you have actually only one lineage is affecting and will split into 2 backward in time, right? In this recombination. And that means that only we scale by beta here. And so I think if you, I'll use that on Wednesday. What is interesting is to compare now the rho divided by theta. So that means we divide the recombination rate by the population mutation rate. So you can see that the 4 NCS vanish. And then we end up with beta r divided by mu. Right? Why is it important? I'll show that on Wednesday. But as a teaser, this beta here makes a difference because if you do that, this rho over theta in species which do not do dormancy, for example, in humans, you end up with r over mu. So this here recombination over population mutation rate is important because it tells you how much mutation against recombination you expect in a genome. And that's, as I will show on Wednesday, very important for doing any inference. And so I'll use that again on Wednesday. Okay, so I think that's, yeah, so that's just an intuition from the model. All those results are true, and that's a key fundamental difference to the second model I show you, is they are true because we have this boundary of m, right, which is the maximum amount of time. What is the maximum time that you can lengthen the coalescent with this? So either, what are the, well, basically I put it in another way, what are the extreme value for beta? So either it's 1, that means there is no dormancy, okay? And what is probably the minimum you can do? Probably 1 divided by m, because that means that every generation you have exactly the same probability, a uniform probability of germinating, which will be 1 divided by m. So if m is really big, imagine it's 100, 1000, you can get a beta which can become very small, so like 1 divided by 1000 or so. Okay, and so that means that your maximum lengthen of the coalescent time is being 1 by beta square. Actually, the maximum you can get, it depends on m square. And so if m is 100, your coalescent time will be lengthened, for example, by 10,000. Right? But you don't get bigger than this here. And that's because we have this boundary on m, which is a finite number. Okay. Okay, so that's the intuition of how it looks like. I use that on Wednesday again. So this is here a classic Kingman coalescent. I did some little scaling here where we have recombination is 0. We have a theta of 1. So we have 3 samples. We have some scaling of the coalescent time and 3 mutations. These are here, this little crosses. If you have assume 1 recombination event, rho is 1, theta is 1. So we have 1 recombination event here. So that you can see this recombination event here, backward in time, and then we find again the same ancestor. And then if you now do with the seed bank, what you see is that, of course, if I put a beta of 0.5 only, which is not very small, but it's only 0.5, that means every seed has 50% chance to germinate every year. That's what it means, concretely speaking. Then you can see that the tree is already lengthened by a factor 4, because we talk in square. And what is also interesting is that as the tree is longer, the recombination being scaled by beta only, so you have a recombination which is scaled by times beta. However, as the tree is lengthened by a square, you have much more recombination event appearing compared to the classic standard coalescent. And it has a lot of property that I will use on Wednesday, right? But I'll show you here the intuition behind the equations. Okay. I hesitate to do this model right now or to do it tomorrow, because I think I would like to spend a bit of time tomorrow on that. Yeah, exactly. So I think I will do that tomorrow, because I think there are a bit of differences and then discuss a bit why it is different. I just want to mention, so basically what is the advantage and the disadvantage of this model, of that one? So the advantage that we have this m boundary, and that means that we assume that the dormancy time cannot be super long, right? And so I think this is probably realistic for many plants, many maybe some fungi, or maybe, for example, invertebrates, so like Daphnia or maybe nematodes, where you could assume that the dormancy cannot be super long compared to the classic generation time, right, or the multiplication time. If you start talking about bacteria, where bacteria can be dormant for hundreds of years, but actually each of this step would be 1 hour, you see that there is a little bit of a problem, right? Because this m will explode, right? So you have to explode this m to, I don't know how big. And this is what so some colleagues have done. So for example, Dario Spano in Warwick and Johan Blatt initially tried, and they said, okay, you know, mathematicians like to do fun stuff. So they say, let's do m tends to infinity, right? And then we put a heavy tail distribution on the germination time. And so instead of having a geometric rate, which of course has a short tail, you can lengthen this time and just assume you have a heavy tail where you're more probable to stay very long dormant, right? And if you do that, the model completely collapses. So you have a problem that it's not possible, it becomes non-Markovian, because you don't recover this Markovian property here of in time that the time to the next coalescent event is nicely distributed, as an exponential is not working any longer. And so that's one of the problem why I think some mathematicians and stochastic people don't like that model too much, because it has this kind of non-scalability to longer times. And the second model I show you is an attempt by these people to solve that problem, and it's a different kind of model which has different assumptions. The advantage, I think, of that model is that I think from a biologist point of view, and I think also practical point of view for species like plants and so on, seems to work really well. We applied it in our tomato, and it seems to at least give us results which we can trust. So I guess that there are two models, and I'll show you the other one, and one should decide what you want depending on the organism you're interested in. So there is no general truth, I think, in here. Okay, and I will stop here for today and continue with this model tomorrow. Thank you. Then welcome to the follow-up from yesterday. And as yesterday, I started discussing about dormancy and seed banks. And so I want to continue and here finish the second type of model, which is commonly used now for modeling dormancy, and get a bit of insights from this model. And if you remember, maybe I still go back to the schematic we had yesterday to recap. So we had this model which is based on an urn model, right? So we have here always a population where we have our samples and the lineages we want to track, and then they can go back in time with this germination rate here, which are this B of I function, right? So that's what I described yesterday. Allows you to say, okay, there are seeds which have different age, which can germinate. And so the main assumption of that model is that the time of dormancy now is bounded by this value M, which is the maximum time that the seeds can stay, right, dormant, or bacteria, or whatever you want, okay? And so the advantage there is also that you can specify any of this function of germination. However, as we have seen, actually, it doesn't really matter because the coalescent and the genealogy of the sample is scaled by anyway the average time that you spend in the dormant phase, and this is this beta value here, right, which tells you on average how long you stay dormant. Okay, so I described that yesterday, and so now I will talk about the second type of model, which was developed by Jochen Blatt group and few other people since a few years. So that's the first version of it from 2013. I think there are since quite a few papers. I will not put them on the Slack because there are too many, but if you want to look for them, search the webpage of Jochen Blatt and you will find the latest ones. And so I will try to recap a little bit several papers together. So the idea of their model is now to use some kind of what is called a structured population, where they assume we have on one hand the active cells, so this is the active population, right? So bacteria which are reproducing, okay? And then we have the dormant cells, right? And so this model is based primarily on bacteria population, where you will have some bacteria which are active and multiplying. And in this here part, or in this population, you have a Wright-Fisher model, right? So bacteria produce daughter, and they die, and you have the classic population model. And in the dormant cell, you have the dormant bacteria, which are quiescent, and they wait until they can resuscitate or get active again, okay? And so we have some reproduction. So this is here the reproduction in the active population, and you have dormancy rate, which is called C. So you can become, an active cell can become quiescent and dormant at rate C. Okay, and you leave the dormant stage and resuscitate or reactivate at rate C times K. Okay, and we can add also mortality. So we can assume that some of the cells here, they die due to other factors, right? So you have natural death because there is a Wright-Fisher model to replace the population, right? But in addition, you can have also mortality due to some other reasons. And also you can add mortality of the cells if you want, right? Of the dormant cells. Okay, and so what is the definitions that they use? I just write maybe a couple of things on the board. Yep. I'm just going to write it. So Kavita is always asking the next question, so it's good. Then I don't have to do so much. Okay, so in this model, basically, we assume that the population size here is the active population. Okay, and that's big N, and it's fixed. And we have big M is the size of the seed bank, right, of the dormant compartment. Okay, and the definition is that N is K times M. So you see this K comes in there, right, because it kind of scales how big is the dormant stage compared to the active part of the population, right? And so the probability of becoming dormant is C over N, because you have to scale it by the population size. So this rate C is scaled by N, in fact, because you become dormant at this rate C, and you scale it by N. And the rate of germination or resuscitation, so that's the dormancy rate, dormancy probability. And here the germination, I'll call it germination, but they call it activation, let's say, for bacteria, is CK over N, right? Okay, and so now the simplification we can do, because otherwise it gets too complicated and too biologically realistic for mathematicians, is to assume there is no more mortality here, right? So basically we have two populations which have constant size, and you only have here active cells which reproduce in the Wright-Fisher model, so the population size is constant. And the dormant cell, you can become dormant, and the dormant cell cannot die of age, right? They just stay dormant until they can resuscitate. You can see now that we have two assumptions which will be implicit here. I mean, which are explicit if I explicit them, but implicitly in the model, the time of dormancy is geometric, because now there is no more function. You cannot tune the function of resuscitation, right? So you cannot say, for example, you have a higher chance of, or you would have some higher chance of resuscitate when you stay 1 or 2 years dormant, or 1 or 2 steps, but you have less later, right? Here it is a geometric function because it depends on this rate C and CK, basically. Right? So you have no choice here. And the time of dormancy here in this dormant population, or the dormant stage, is potentially unbounded. So if you have no death, potentially you can stay dormant for a very long time, right? Depends on your probability that you germinate or that you resuscitate or not. Okay, but nothing, there is no constraint from the biology point of view to say you should not be more dormant longer than X time, right? This is not possible here. There is no function. Of course, this how long you stay dormant on average will depend on this rate, right? Obviously, but potentially the variance can get really big and can get dormant for a very long time. Okay, and so how the model is built. So I have a little graph. Maybe I'll show this graph first. So the model is built really as it's kind of a 2-island model where if you know a bit of population genetics, and I think last week you had some little spatial structure courses, this is exactly how it's built. You have one island, which is the active, which here they call the plants. So it is very confusing, all these papers, right? They use different terms all the time. So I'm trying to be consistent, but when you read them, don't worry. That happens all the time. So this is the active population where we have the Wright-Fisher. And on the other hand, we have here the dormant stage. And you can see you have, for example, here this individual has 2 offspring. One of the offspring gets to the dormant phase, stays dormant for a while, comes back in the active population again. But that one, for example, did not leave any offspring further, right? Okay. And what you can do, so how they build the model basically in a coalescent framework is to define now blocks of lineages. So you have either lineages that you track from your sample in the present into the past, right? And so if you track these lineages, either these lineages, they fall in a block of lineage which are in the active part of the population, or the lineages can be in the dormant part. Okay, and so if you do mathematics and stochastics, what you can do is you can track these different blocks of lineages separately, because different things happen to them, right? So what happens to the block of lineages? So the lineages which are in the dormant stage, nothing happens to them. They just stay there basically for a very long time until they resuscitate backward in time, right? Or until they go back to the active population backward in time. But the block of lineages which are in the active population can merge by coalescence, because the coalescence is only allowed in here, in the active part of the population, obviously. Okay? So that's the intuition behind it. Okay? So I will just skip. I mean, if you want to look at the stochastic and the mathematics, you can look at the paper and we can discuss about them in the next days if you're interested in the techniques. I just want to write here how you count and what is the transition of the Markov chain that allows you to do that, what are the different states. So what you're doing is you assume we have n lineages in the active population and little m in the dormant population, okay? And then how you change this to the transition to the next state is either you transition to n-1 m. That means here we have a coalescent event in the active population. So you lose a lineage in the active population, but in the dormant population it stays the same. And this occurs at rate choose n in 2. So that's the usual coalescent in the active population. Then we can have a transition where we now lose one lineage in the active population because it becomes dormant, right? And it goes into the dormant population. And that happens at rate, so C n, because you have n lineages here, and C is the rate, and you have the other way around, that we have, you move one lineage from the dormant to the active population. Okay, so that's the transition of the Markov process of the coalescent in this model, basically. Okay, does that make sense? I think it's just intuitive. I will stop here the math, right? Because afterwards it gets the demonstration of the Markov process and how you obtain the process is getting too complicated, or at least for my taste. But that's the intuition. And so what happens is you generate such coalescent tree where basically you can generate the coalescence of the samples, and you can see that now what they do is that you have some lineages. They put in dotted here, that's the time that this lineage has spent into the dormant stage, right? And you can see that the difference to the model we have done yesterday is basically now that this time is non-negligible compared to the coalescent times, right? Potentially this can be very long. And before yesterday, if we were to do the same graph, all the dotted would be just so small in the graph that you will not really see them, because you have this M window, which is maximum time you can stay dormant is much more shorter, right? So here now you can see that you have lineages which alternate between being active, then dormant, then active again, and then you have coalescent only, of course, in the active part. Okay, so you can build now a coalescent process, and you can build a coalescent tree and estimate many different statistics from it. Some of them we will see with June in the next lecture, so I will not talk about them, but we can do all the classics, diversity and all measurements, right? What I would like to write is one of the results that they obtain, which I think is from here interesting. So that's, they calculate the time to the most recent common ancestor of this this process. And they find that in the limit, so you always have to take the limit when the number of lineages tends to infinity. And you find that this is the time to the most recent common ancestor. So it's 1/2/k plus 1/k squared. So if you look at this and you look at that, you might think already there are a couple of little weird things going on here, right? So what is maybe a little bit weird in this formula? Or what is maybe not weird about it? Let's put it that way. Start with the positive. So does it kind of make sense? So what would you expect for the time to the most recent common ancestor? Should it be short or long, depending on k? Okay, so if k is 0, right? Let's do it easy. Okay, you have already an issue, right? But let's say it's very small, right? So what's happening if k is very small? Here. What does that mean, biologically speaking? Okay, so you wake up at a very slow rate, right? So that means you can stay dormant for a very long time, right? So when k tends to 0, right? Not 0, but 10 towards 0, then you lengthen the time of dormancy. So what should you expect for the time to the most recent common ancestor? Should increase, right? Because you stay long dormant, so you take much longer until you find a common ancestor, because those times will get longer and longer, right? So is that really true? That kind of makes sense, because now you see if k becomes 0 and close to 0, this number can become very big, right? So that's okay-ish, right? Now, what is the little bit maybe surprising part of this formula? What is the rate that is not there? You have this rate c. So this is not here. So it doesn't influence apparently whatsoever the time to the most recent common ancestor. So this rate of the rate at which you become dormant, and also it is in the reactivation rate scaled by k, but nonetheless it is there. Obviously, that doesn't seem to influence whatsoever the time to the most recent common ancestor. I must admit, I still entirely don't understand this result. So if you have any idea you want to discuss, I'm happy, because I will show you a couple of the result, what it means. They provide in this paper here some expected time to the most recent common ancestor, right? So that's the expectation under this model for different size k, sample size n, and dormancy rate of in this rate c. Okay, and you see d is 0, that's the mortality, and so that's the model I showed you. Okay, so if we look at this table, what do we see? When k is very small, so 0.01, right? Whatever c is, you find that if you have a sample size, let's focus on sample size 2, we find that the time to the most common ancestor is 1.02 times 10 to the power 4. Right? Be careful, because here, this is multiplied by 10 to the power 4 in this part of the table. So that's very long, right? Makes sense because k is very small, so you get it from this formula, basically. You can see that indeed c does not influence whatsoever here, the rate. Right? Now you move to a higher sample size. If we have more sample size, you can see that the time, of course, increases, right? And you have an effect now of c, which has an effect. So the higher the sample size, obviously, c has an effect on this time, which is obviously not the case for this calculation here of the TMRCA, because here they do it for 2 and they do it for n lineages and they find the same limit. So obviously there is a little problem here in the approximation that when they calculate the limit of this time to the MRCA for higher sample size, obviously there is a problem somewhere that the approximation doesn't work really well. Right? So it works for sample size 2, but once you have higher sample size, there is some limit which obviously some terms should vanish, which should not vanish. Okay, so that's the first thing that we can see. Now you can compare this number to k is 1, and you can see that the numbers are way smaller, right? So here we talk about in 10 to the power 4, here we are talking about 4. So you see that now here we have a huge scaling effect of the k value, and that kind of makes sense because this k, again, is at the square here. Right? So basically when you have a very small k value, which is here 10 to the power minus 2, you explode the time to the most recent common ancestor in the square, which is kind of consistent actually with the model that I showed you yesterday. So that's kind of the interesting thing, is that in a way, at least from the aspect of the model, it is consistent that you get again this square scaling, because you have 2 lineages have to find a common ancestor at some point in time, so they both have to germinate. And that is true for both models, right? In this model is also true. You need, if you want 2 lineages to be there, either one has to be already in the active population and the other one dormant. But if both are dormant, they also need to both germinate in the same time, again. And you can see that when you go to k is high, that means that now the population, if k is very big, the dormant population is very huge, right? Compared to the population size, sorry, is very big compared to the dormant population. And so that means that then the dormant population has very little influence, and we almost go back towards the classic Kingman coalescent, actually, because the dormant population is so small that it has almost no influence any longer on the dynamic. Sorry? Yeah, so this is by recursion. So this is by recursion and a numerical calculation. Yeah.
So there you see it's dependent on C.
Yes. So there, I mean, you see here, I mean, here not so much, but here it depends on C, obviously. When you look, the C value are here, and you can see that if you change C from very small to very big, it is affecting here the value of the TMRCA. And not for 2, for the sample size 2, but then once you have more sample size. So I'm happy that we can re-go through the paper if people want to understand better, but there is still a lot of mystery after many papers I read from these people to me. So I think if you want to dig into this, there is a lot to understand, I think. Okay. So there are some aspects which I think make sense, and we kind of fall back on this square scaling that we had yesterday, right? And so that's kind of at least reassuring. But there are some other aspects which are a little bit more difficult, and especially to handle the stochastic in this model is not as easy, I think, as I would argue, as the one from yesterday. Okay, and so what is the issue? Oh no, so what do you, sorry. Yep.
So that calculation is approximate?
Yes, it's an approximation, yeah. Yes, yeah, so. Yeah, so there is still a C, and then I think when they do the approximation to calculate the TMRCA, then the C vanishes. Where it is, I can't remember because there are several papers where it's done, so I would have to look again where exactly they do it. Yeah, yeah. So maybe I should let's be more precise and write here. So approximately. So this is not. What you can also calculate, and I think that's also interesting if you analyze a bit of data, so I will not give much detail on the side frequency spectrum because June will do more in the next courses, but what you can calculate is the allele frequencies that you would expect based on the coalescent tree. So you throw mutation on your coalescent tree, and if you have mutation on the tree, you can calculate their frequency in the population, right? So that is called the side frequency spectrum. I think you did it last week already, so I will not go, I don't need to give more details. And so what they do here, so the bottom graph is the graph from our own paper where we use this model I showed you yesterday. So that's called what is called usually the weak seed bank effect because it has this boundary of time, of dormancy. And you can see that the side frequency spectrum you obtain, this in black is the Kingman coalescent and the white one is the one with the seed bank, and it's exactly the same when you scale everything in relative. In absolute number, we get much more diversity in our model than in the Kingman coalescent because the coalescent tree is much longer, right? Because you maintain diversity in the seed bank, so the seed bank is generating much more diversity than expected under the Kingman coalescent, right? But the side frequency spectrum in relative, that means the frequency in relation to one another, are the same. However, when you take now this model with the true island model that you get, this is the black one here, this is the gray is the Kingman, and the gray, yeah, so actually that's, so the gray is the third bar, right? So it's this one, basically. So it's not really clear, but you can see this is a decreasing, right? It's a monotonously decreasing function, the SFS in the Kingman coalescent. But I hope it's clear that when you see the black bars, the first one, they have a U-shape side frequency spectrum. So now you get a completely different distribution of the allele frequency in this model. And so that basically because we have this long time to the most recent common ancestor, but also the branch length in the tree and the relative length of the branches in the tree is massively affected by the strong dormancy compartment. And especially if K is very small, then you indeed end up with very long branches. And what you get is actually very long internal branches in the tree. Right? So because these branches are proportionally much longer than expected under the Kingman, and then so you get a completely distorted tree, and you get this U-shaped SFS. So one way that they propose in this paper, that's the paper I put on Slack, where I took this graph from, this two. What they propose is first they compare that to what you get into a two-island model. So if you remember the simple island model you did last week, you can compare these results. And of course, it's not exactly the same because in a two-island model, you have coalescence in both populations, right? So that's a classic model where you have two populations with migration. And in both populations, there is the Wright-Fisher model. Here, one of them doesn't have it because you're dormant. Right? So that's a model is similar, but not exactly the same. So that's interesting. And the second part, which is of course interesting, is that they show that here so that's the SFS in absolute number. Here on the right is the normalized SFS. When you normalize, and you can see that compared, for example, to the Kingman, you have much less low frequency variant. However, you have much higher high frequency variant. Okay, so the SFS is really distorted compared to what you would expect. The intuition from the people, so Jochen and other people, is to use this SFS to test whether there is dormancy, for example, in bacteria population. So how would you do that? You sequence, I don't know, some bacteria strains, right? So many strains of the same species. You build a side frequency spectrum, and you try to understand if the frequency spectrum looks like U-shaped, maybe it is due to indeed dormancy, and you would be able to test whether there is dormancy or not. Sorry? Yeah, so I mean, basically the ratio of the different branches is completely different from the Kingman. So what you get is much less shorter coalescent. So maybe I do another graph of the coalescent, which is a little bit more true. So I think the one they have in the paper is maybe not the best. So what you would observe in that model is that in the active population, you have very fast coalescence. So that will be in the active part of the population. And then if this lineage becomes dormant, this can be dormant for a very long time, right? Basically. And then you have the same, that you have maybe some other individuals which coalesce very fast here. And then you have now these very long branches again. And then you have the same, if you have another sample here, where we might have even a completely mashed here, then maybe we have something like this, and then maybe here, right? And then these branches can be very long here, right? And so you have a complete time scale separation between what's happening in the active population, which is very fast coalescence. And then you have to wait now a long time, potentially, for all the lineages which are dormant to wake up and then end up in the active population and then coalesce. Maybe I should not put this here because I did the dormant lineages, but let's put it like this, right? So I think this tree is a little bit misleading, I would say, because it doesn't really reflect this kind of distortion of time, right? So you have really a time scale difference, and I think the whole model of the Markov chain is based on the fact that you have this time scale argument separation of time scale between a short time scale here of fast coalescent in the active population, which narrows down actually many lineages to very few. And then you have these coalescent times, which can get very long for the last remaining lineages until they coalesce together. And so I think this tree is not exactly, I would say, is maybe not the best representation you can get for the intuition. And so you can see that this will distort completely the SFS, right? Now.
So I was thinking in terms of, because we get SFS for selection also, positive selection. So is there a connection?
So yeah, so the connection is in a way that if you take the timescale argument, that's exactly the point, is that you would say, well, there are very many lineages which can coalesce very fast, and that looks like a star-like genealogy, right, at the bottom of the tree. But then you have the other remaining of the coalescent have to coalesce long time back. And imagine you have bacteria, right? So if you take bacteria, they reproduce within an hour. So you have a very fast set of generations at the beginning where you have a lot of coalescence. But then if they fall dormant for 100 years, you have to wait. Right? But there cannot be many lineages which are dormant for 100 years, basically. And so that's exactly the point, that it looks like a kind of a positive selection. It's really not, right? It looks like it because you have this star-like genealogy at the bottom of the tree in the recent times. And so the time scale effect here is very big, potentially. So one of the issue is, so if I don't want to discuss about this. I think if people are interested, we can have an extra session to discuss what that means. The point is that if you want to use this FS to test for seed bank, I think you run exactly into the question from Kavita, is how are you sure that this is not due to selection, right? So you can revert the question, is that if you see this SFS in your data, it doesn't mean you have necessarily dormancy. It can also mean that this is due to other factors, especially selection can create exactly the same signatures. And so I think that's one of the key questions in this model, is how you interpret it in the light of the data. And I think there is a lot here to do. If you're interested by these problems, I think that for sure will be an interesting question. I'm just mentioning this question because if you're interested, for example, in bacteria, which are human pathogens, obviously you would like to know, is this selection or is it dormancy, right? Or both, which is even worse, right? Okay, and what I would like to highlight now is that there is one or two peculiarities in this model. And I just copied that from this paper to leave you reading it quietly. I will just sum it up in a sentence. Sorry. The main point is that this model, and so one of the feature of the Kingman coalescent is that if you take this limit into the number of lineages, little n, or the samples tending to infinity, which is how we derive many of the statistics from it, you're sure that there will be a most recent common ancestor in a finite amount of time. Okay, so this is what this property, which is called coming down from infinity, which I think for biologists makes totally no sense. So it took me ages to understand what it means. Basically, the idea is that you have many lineages, and at some point you know that under this process you will come down to a finite amount of lineages which will coalesce. Right? And I think this is our intuition as a biologist. So here, if I'm really a biologist, you kind of know at some point intuitively that there must have been common ancestor to most of the species, right? To the bacteria also have a common ancestor. Maybe it's a little bit complicated, you have several ones, but it can't have been billions of bacteria from the beginning of times, obviously, right? I mean, think about all the species we know there is some kind of common ancestor at some point to different species. Now, the problem of this model is that it does not come down from infinity, as the mathematicians would say, which means that if you start with an infinite amount of lineages, you're not sure that there will be a common ancestor. It's not guaranteed. So that's weird, right? So what that means, I will explain it in other words, what that means practically speaking, and that explains part of the problem when you see this calculation of the time to the most recent common ancestor, is that if you take 10 samples, you do your analysis, you see indeed they coalesce to a common ancestor, right? You take 20 samples, you find a completely different common ancestor. You take 100 samples, you find a completely different common ancestor, or you don't even find any, right? So that is, for me, a mystery. I must admit, that reaches the limit of biology and mathematics here, which do not agree with each other, right? Because clearly, there is some fundamental process here in this Markov process that violates our biological intuition, in my opinion. And how you reconcile this, I think that's a great question, I think, to understand how we could apply this model, right? Because you see, if I apply this model to data and I don't get the same answer if I increase my sample size or decrease it, that's going to get very difficult to do in the interpretation, right?
Because the dormancy time can be very long.
Because the dormancy time can be very long, yeah.
M generation, it has to.
So, yeah.
So does that make answers?
I think the question would be, I guess one of the only ways that you would have to bound this time of dormancy, right? So we have to say, okay, you have this geometric function of dormancy, but it has to be bounded by something at some point. And then you're sure that then you collapse again to a model which is well-defined and will come down from infinity, because then you must find a common ancestor, right?
Has that been shown?
This has not been shown. So I think the mathematicians don't like to bound stuff. I think you probably guess. And so I think there they have this dormancy. So they would tell you the time of dormancy is on the order, of course, of K. But of course, you can have very, 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 very long time of dormancy potentially. So I think if some people are interested to make this model more applicable, I think it would be great. So just as a little hint. But I think that's an interesting question. So in a way, it's a bit difficult when you see this side frequency spectrum. It's very promising, right? You think, okay, we could do something with it. But then if the model is not consistent when you analyze it, it's also a little bit difficult, right? So I think I just want to point out that this is a field which is, I would say, fairly recent, right? There are a few people working on there, developing a lot of models. And so I think it's very exciting also to see, science sometimes progressing, but then you see the problems, right? And to reconcile biology and mathematics is actually here very interesting in this case. Okay, are there any questions about this? Okay, so that's a little cartoon I took from this paper. I put you on the Slack. So this is just a recap of, that's a Kingman coalescent genealogy. So that's the classic one we talked about yesterday and June talked about. That's the one with the weak seed bank. So the weak seed bank is called weak because you bound it, and potentially the effect is, of course, not so strong, right? And then this is the effect of the strong seed bank, so the model I just showed you before. And you can see that here this is also misleading because actually this could be way, way longer, right? And so it looks like, oh, it's not much different from that one, but that's not true. Because if you take K very small, your tree is really exploding, right? Potentially. And the group of Jochen Blatt, they did another model, which is called also the strong seed bank with the simultaneous switching. That's this one here, where they assume that sometimes you have some cues in your environment where all the bacteria germinate, or 90% of them will get activated, because I don't know, there is sugar or something. So they all get out of dormancy and start multiplying. And in that case, you can see here that so at that stage, for example, here, several lineages get up when there is the dotted line, and several lineages just resuscitate at the same moment. Right? Under the classic model here, this is not the case. They can stay dormant a different amount of time. That's a stochastic time. Right? But when you have the simultaneous switching, then several lineages will resuscitate and get active in the same time, because there is at that moment maybe more nutrients or something. Right? So that's create again some differences in the model. And if you read the end of this sentence here, this is exactly what they say. In the simultaneous switching model, you can have all sorts of behaviors. So either it stays infinite, you never find a common ancestor. Either it comes down from infinity, so you find one, or it comes down from infinity after a finite amount of time. So it depends on the choice of parameter and initial conditions. So that's not so trivial what can happen in this kind of models. Right? So probably that's also realistic for bacteria, that you could have such effect where many bacteria simply will get active. But obviously, what are these parameters in these conditions remains, I think, to be explored a little bit better now. Okay, are there any questions, remarks? Beside thinking that all these models are useless? Yeah. No, so here there is no environmental change, nothing, right? So we just assume you have an environment and then some will go dormant, some not. I think that's exactly the point, is that in bacteria, a lot of this dormancy also happens based on the environment. For example, you treat with antibiotics, many of the bacteria will start falling dormant, right? So it's not entirely a random switching to dormancy, right? So these parameters can be tuned. You could tune them in simulations to say, maybe something happens, many bacteria fall dormant, then they wake up again, right? So this is not accounted here, right? This is a pure stochastic model where the dormancy is a stochastic process here, yeah. And so that probably saves you, right? In a way, the biology, probably if you do that, it probably kind of save the model to make it more realistic, and then probably again, you fall down on some realistic behavior. But mathematically, this is much less interesting, obviously. So yeah, I think one probably should do it by simulation. Yeah. Okay, so I put a little recap of yesterday and today, just for you also to remember some things what we talked about. I hope I convince you that dormancy is very common. And I just mentioned here some extension if you're interested of a couple of recent papers now which have been done. So I have a postdoc who has worked in Paris on working on plants in urban habitats. So if you go, for example, in Paris, you have all these trees, right, along the street. And actually, all the plants, especially many of the small plants which live on these patches, they do seed bank. Because of course you get cut all the time, right? Or you get, you have dogs making caca everywhere. You have all the people cutting plants randomly, and many things happen. So actually, this plant, that's how they survive. They do a lot of the seed bank so that they always have seeds which germinate at some point in time to find the patch empty. And so it's kind of an interesting, very micro little seed bank adaptation. And you can do a lot of stochastic with this because you can just survey all the plants, right? You take a street and you follow like hundreds and hundreds of patches, and then you can track this plant over time. And then you have also a lot of spatial system with the meta population, which are done by Frank de Hollander, a mathematician, but also some people in Montpellier who worked on extending this model to meta population, especially for plants, but also for many fungi and bacteria and so on. This might be relevant because you always have different populations. So I was thinking about the microbiome. That's exactly the point. If you have the spatial structure, you have different parts of microbiomes which are not always connected, right, with each other really well. And I think it opens a lot of questions also that we try. I will talk about it on tomorrow. For example, would you plan to dormancy or selfing? I talked with someone about this yesterday a little bit. That's one of the key questions of this kind of life history trait seems to affect a lot how plant or animal or fungi can adapt to a lot of different situations. Would you do dormancy or dispersal, right? So are you preferring to disperse in time? That's dormancy. You put all you put your eggs in different baskets in time, or do you disperse in space rather and try to colonize new habitat to avoid extinction all the time? And so these are kind of key questions in ecology. If we zoom out a little bit from population genetics to explain how most of the species on the planet, especially plant species, behave. Okay, I will stop here for this one and move on to the second part. Unless, yeah.
So dormancy, like it's about the length of time it's staying in the setback. Bringing out, trying to make an analogy with this person. So what would it be like? This person can be two things. Either it's the physical distance that you are dispersing, or like you produce, like you have high fecundity, so in general you have a high dispersion propensity to reach these cells.
So I think it's rather the first one. So the question is, imagine I'm a plant and I produce 100 seeds, right? Should I invest that the seeds stay in the same place, but maybe do dormancy so that they will wake up at a different point in time when the conditions might get better in the future, right? Or do I disperse all my seeds and do very tiny seeds which can be moved by the wind or whatever, by animals or something, and then I hope that they will reach a patch or a place where they will be also a suitable habitat? Or do you do a mix? Do you say some will be heavy and stay here, and some will be light and disperse? Right? And that's actually a trade-off which is very common for many plants, but also many animals. So marine animals, for example, have exactly the same issue, right? So do you stay in the same place, or do you colonize different places? And dormancy versus dispersal is a huge topic in ecology because of that. Yeah.